Hi, my name's Iain Hagan and I'm going to talk to you about the PhD position that we have available in the Cell Division group here in the Cancer Research UK Manchester Institute. So the project is to study protein phosphatases, and protein phosphorylation is one of the major ways by which the behaviour of proteins is changed in signalling pathways to alter the fate of cells. So consequently, because of this massive impact of protein phosphorylation on cell function, the study of protein kinases has really dominated cell development, cancer biology for several decades. The study of the phosphatases that remove the phosphate, and the removal of the phosphate is equally as important as putting it on in the first place, that has lagged behind the study of the kinases. Now, the major reason for this is that it's really important to have a specific phosphorylated substrate for a specific phosphatase to understand how that phosphatase works. Protein phosphatase 1 and protein phosphatase 2A account for over 90% of the phosphatase activity that's directed towards phosphorylated serine and threonine in a human cell. It's therefore really important to understand how these phosphatases work and how they are controlled. A couple of years ago we published a paper in which we described how protein phosphatase 1 regulates protein phosphatase 2A by activating it to drive cells out of division. Now, one component of this analysis was it looking at a very well understood phosphorylation event in the end at the C-terminus of protein phosphatase 1. This site had previously been shown to inhibit the activity of PP1, and sure enough we found that it did. However, we found an additional property of phosphorylation on this site. We found that it targeted the protein for destruction. Now, because protein phosphatase 1 has over 200 partners and many more substrates, this mode of control is really important for protein phosphatase 1 function, and will have major impacts on cell behaviour. Working in the lab, this project will exploit the fission yeast, Saccharomyces pombe, as a model organism to address PP1 function. Fission yeast is ideally suited for this because it has a limited phosphatase repertoire. However, all of the core processes of cellular function are completely conserved through to humans. So the project will have three principal components. In the first, we want to understand how phosphorylation in the C-terminus is processed by the cell to target the molecule for destruction. The second component exploits the fact that there are two different PP1 isoforms in fission yeast. The first is regulated in the way I've described. Phosphorylation C-terminus inhibits and targets for destruction. The second, a molecule called SGS21, doesn't have this site of phosphorylation, yet it has another site that we know is phosphorylated, yet we don't know what it does. Does it inhibit it? Does it target for destruction? We'll look at that. The third component of the project is a very straightforward structure and function analysis. We'll map phosphosites in the two isoforms, mutate them, and determine what the impact of phosphorylation on these residues is. All of the technologies for studying yeast and conducting protein phosphatase assays are established within the lab. We also have pipelines for mapping phosphorylation sites on any molecule we want, in this case protein phosphatase 1. There's 4 other members of the team are studying protein phosphatases in both yeast and in human cells, and there's an additional 4 lab members who are using fission yeast to study cell cycle control. It's therefore a really dynamic and active environment, and all of these activities will be taking place in a highly vibrant institute, so it is an ideal opportunity for a really great PhD.